Hi, everybody. Very happy to be here at this conference and with my colleagues and friends in longevity. My name's Yuta. I'm the founder and CEO of Accelerated Biosciences. We're based in Philadelphia. I guess I have to stay here. We're based in Philadelphia, and we're working in cell and gene therapy. So today, my job is really just to get everybody here up to date on what's happening in our industry. So as a show of hands, who here is actually working in cell and gene therapy? Anybody? All right, we got some people. All right, very good. Okay, so these guys are the experts. So those of you without your hands up, they're the ones to talk to at cocktail hour later today. All right, if you have a drink, talk to them. You'll find something very interesting, I promise. Okay, and for those of you with your hands already up, I have to apologize because for this session, I'm going to deliberately and violently simplify all the work that we're doing so that we can get everybody up to speed. And I promise to leave no one behind. Okay? All right, so we're going to go through some of the basics of cell and gene therapy. I'm going to give you a lot of statistics and data on market conditions and where we are at today. And I will talk a little bit about how I'm contributing to the process. Okay? And there's a lot of data here, a lot of charts. Focus. You don't have to take pictures of them. There's going to be a QR code at the end. If you take a picture of that or scan the QR code and email me, I promise you I will send you the slides. Okay? Okay, so when I think of escape velocity, this is the image that I conjure up in my head. Which begs the question, am I going to make it? I'm chronologically 52 years old today. I don't know. That comes dangerously close to some predictions of when we will hit escape velocity. What, 10, 15 years? Right, Aubrey? So very, very close. And you know what? My father has gone through 2 bouts of liver cancer, and my mother a bout of colon cancer. Wow, that sucks. So am I going to make it? Don't know. That's why I'm here. I'm here to talk to colleagues, to work with my colleagues, and to really push the science in cell and gene therapy and longevity to see if I can buy some more time, as Aubrey just said. I'm predisposed to these cancers. And I'm sure a lot of you in the audience has the same experience as me, right? But this should be the incentive for us to wake up every day and really push hard on the science. Now, a lot of people are working on prevention, whether it's getting enough sleep, dietary supplements. It's a lot of different ways to come at it, right? But all I'm saying is pick your path, pick your passion, and just push, because it'll take everybody here to come together with prevention and working on the back end to solve problems to reach that longevity. So I picked my passion, and it's cell and gene therapy, right? So cell and gene therapy really is developed to solve problems when the problems have already happened, okay? Parkinson's disease, diabetes, a lot of the diseases we can attribute to aging, we can probably solve with cell and gene therapy and really, really solve the root of the cause. Some basics. Cell therapy is when you take a cell and you replace a healthy cell and replace or repair a damaged one. Now, when you look in the internet, you'll get a picture like this, which I think is way too confusing, right? So I'm going to simplify it for you a little bit. You're basically taking cell and you're making it either kill or repair, right? So you can take an immune cell, like a T cell, an NK cell, or a macrophage, and you can set it against a cancer, which we call immuno-oncology, or you can now use it against a senescent cell for senolytics. On the repair side, you're taking a cell of the same type to replace a cell that's gone bad. So beta islet cells for diabetes, for example, a neuron for for Parkinson's, for example, okay? So just remember that at cocktail hour. Kill or repair, pretty simple. Another takeaway concept in cell and gene therapy is autologous and allogeneic. Autologous is when you take your own cells, process it, and repair your own body. Allogeneic is when you take a healthy donor cells, mass produce it so that everybody can use it, right? Some pros and cons. For autologous, no rejection because it's your own, okay? But the problem is, it's a service model. Every patient has to come in, have your cells manufactured just for you so you can be applied, right? So this is a hospital model, right? So with allogeneics, if you can find a healthy cell, you can mass produce it, which means the dose cost goes down dramatically, right? And then you can distribute it and everybody can use it. But the downside is you have to overcome the immune rejection issue. Okay, so there are no free lunch. In gene therapy, very, very complicated, but extremely popular and very, very successful these days. You're inserting or replacing a functional gene for the non-functional gene. Something's gone wrong, mutation, or you were born with a hereditary disease. All we're doing is taking something that's functional and putting it in there to replace it. It could save your life. Okay. You may have also heard of CAR T therapies. It stands for chimeric antigen receptor T cells, and the T stands for thymus, which is an organ right above your heart, between your lungs, right here, okay? And that's where your T cells are activated. That's why they call it CAR T, right? And so basically what's happening is, your immune cells have lost its ability to recognize the tumors flying around in your body. So all we're really doing is taking that immune cell, a T cell in this respect, right, and mounting on a chimeric antigen receptor, a CAR, which is CD19, and growing it out and putting it all back in your body, literally telling it to say, if you see anything with a CD19, capture it and kill it. And it's been extremely successful for the last 10 years. Okay. So since 2017, more than 34,000 patients have had this therapy. Okay. Recently, the FDA have flagged 20 cases of secondary tumors, which means the therapy actually caused another tumor after they were in remission. But the numbers are so small, it's minuscule. The cost benefit is still extremely high. Okay, and at Mass General they did a study to aggregate all the different CAR T therapies, and the conclusion was 76% remission and 33% some sort of toxicity, neurotoxicity or cytokine release syndrome, which is basically just kind of almost like an allergic reaction to the therapy. Okay. All right, here are the industry updates. Lots of data here, so like I said, focus. I will send you the slides. As of today, the FDA has approved 41 cell and gene therapies. Okay? And I'd like you to note, in 2024 alone, 7, right? Which is the same number as the last 2 years, right? The whole year, right? So we have more to go. And in Europe, 19. And I'd like you to note in Europe, the little purple stars above the bars. I'll explain that in a little bit. So these are 15 really anticipated cell and gene therapy products that we anticipate will pass the FDA this year, okay? And as you can see, we already have 7 of them. That's pretty exciting. And this is how it splits out. 41 approved therapies, 22 of them are cell therapies, 10 of them are gene therapies, and 9 of them are CAR T therapies. And the various indications you're going after, mostly hematology-based, and hematology-based is including blood cancers or leukemias, okay? With Europe, they passed 19 cell and gene therapies to date, but unfortunately, 7 of them over the last few years have been withdrawn or discontinued. So they have 12 available to be used in Europe. Now with this, I wanted to share a story. There's a product called Glybera that was put out by Unicure back in 2012. And that was, at the time, the most expensive gene therapy on earth, €1 million per therapy, okay? And no one would pay for it. The governments wouldn't pay for it. The insurance companies wouldn't pay for it. So what happened was there was a doctor in Germany that really pushed for it and had 1 patient treated, where the government and the insurance company pitched in and paid for it, right? And that was very, very successful. But after that, no one wanted to pay. And so basically what happened was they had 3 therapies left, and they gave it to the next 3 patients for €1 each. That was it, and they withdrew from the marketplace. Okay, so as you can see, between the US and Europe, it's a little harder over here to get things passed and get things paid for, right? So the US is still going to be the most, I guess, promising market for new therapies. Okay, the letters are really small, but here are all of the cell and gene therapies that have been approved in the US by the FDA since 2017, which is the beginning of the CAR T therapies. All right, can you spot the most expensive therapy on earth? Can you see it? All right, well, the winner comes in near the bottom, 5 from the bottom, right? Orchard Therapeutics, Lemneldi at $4.25 million. Whew, wow, that's pretty scary, right? And it's to treat metachromatic leukodystrophy, or MLD, which affects children mostly. And if you have this, that means you're lacking an enzyme to process some fats, and it accumulates in your brain and your nervous system, and you will die by the age of 5. And so the question is, what if your kid had it? Would you pay for it? I certainly would. Can you pay for it? Probably not. We got to figure out a way, right? I want you to zoom out now, and I want to show you a different perspective. I picked 3 different samples here: hemophilia A, sickle cell disease, and thalassemia, okay? The red number is how much it costs to keep 1 patient alive throughout their lifetime. The blue is the gene therapy one-time cost. Now, if you look at it from this point of view, all of a sudden, it makes a heck of a lot of sense, doesn't it? Right? For governments, it's like, well, wait a minute. The cost-benefit makes perfect sense. Now all I see is a financing problem, right? So for all of you out there working in Web3 or FinTech, I think this is something interesting to work on, right? So I believe that the future lies somewhere in between something like what we do with financing mortgages for a house, right? Can we get that future payment to apply to this? Don't know. But I think it can work, right? And over the last decade, a lot of new companies have come out, right? In 2023 alone, there are now 2,700 plus companies developing cell and gene therapies, right? Usually this is a function of new technologies that are coming on and people financing into this industry because they're starting to see cures like we are seeing with the gene therapies and cell therapies that are being approved, okay? And this is by region, and I'd like you to note that the largest region is still North America at 42% or 43%, but surprisingly, Asia Pacific is blowing past Europe at 33%. A lot more activity over there. And I travel there quite a bit. A lot of activity in Korea, a lot of activity in Singapore. So if you're traveling through Asia and you're trying to raise money, those are the places to go. A lot of clinical trials as well. 2021, 2022 were the peaks because those were also the peaks of the financing periods. And since then, we've been kind of going through a nuclear winter for biotech. So very, very difficult to raise funds in the last 2 years. And you could probably see in the news every day that biotechs are laying off people on a daily basis, right? But I believe that we're about to turn that around. And that's a function of stability in inflation. That's a function of political stability, depending on where you're at, right? And yeah, and I believe that all the dry powder, which accounted for about 300 billion or so in biotech, needs to be deployed. Otherwise, just sitting in the sidelines. And I'm sure the LPs are going to be very angry that they're not making their money back, right? And this is how it splits out by phase. Still a lot more in phase 1, moving all the way out to phase 3. But as I want you to note, 112 phase 3s, that's quite a bit. That's quite a bit, right? All we need is a small percentage of that go through, and every year we'll have more products coming through. Right? I found something really interesting I just wanted to mention to everybody. Turns out that if you develop an orphan gene therapy, there's 3.5x more likeliness to be approved, right? This is once you enter phase 1 clinical trials, right? Compared to average drugs. So in phase 1, 48% higher, phase 2, 65% higher, phase 3, 30% higher. So orphan diseases are defined as diseases where there are less than 200,000 people in the world. And because there's so little patient population, the biotechs and pharmas have no incentive to really spend the money to develop drugs, right? And so what the FDA has done is put in a lot of incentives to say, hey, if you get something passed, you get 7 years monopoly, market monopoly over it, that's it. And you get a lot of tax breaks, et cetera, et cetera, et cetera. So some people are chasing down this path. Here are the financings, and as you can see, after 2021, everything kind of just falls off a cliff, right? But I believe we're in a recovery. I think 2025 is going to be the year everything comes back. So, what do we see? Gene and cell therapy really is at an inflection point. We're going to see more approvals, more startups, and more investments. And these are actual cures that you just pay once and you're done. Incredible, right? This is how we buy time, all right? So what am I doing? How am I contributing to this? So I run a company called Accelerated Biosciences, and we have a platform called the Human Trophoblast Stem Cell. The biggest problem today in cell therapy is that it's very, very hard to find the right starting stem cell source. You have ethical issues, inconsistent cell source, you have to choose between scalability and immunogenicity, and many times the IP is unclear and the CMC is put together the wrong way or very lacking. We've actually taken care of all these issues, right? And this is a timeline of where all stem cells come from. And on the left there, you can see first 8 weeks is embryonic stage. From 8 weeks to birth event is the fetal stage. And then you have the birth event, and everything after is considered adult. Okay? So human embryonic stem cells are taken from leftover embryos at IVF clinics. But when you use them, there's a lot of ethical issues. A lot of people don't want to touch that. Okay? And then in the middle, during the fetal period, you can get a lot of cells from aborted fetuses. Also quite controversial. Then you can get to the birth event. That's when you can really source a lot of stuff. Cord blood, placenta, amnion cells, right? Wharton's jelly. And then beyond that, it's from adults. In your bodies, there are a lot of stem cells. Adipose, bone marrow, for example. Okay? But typically, the more to the right you go, the less active the cells are, the less ability it has to scale. But when you move to the left, the more active they are. Unfortunately, they're very hard to get. But we were very, very lucky to be able to source our cells from an ectopic pregnancy, which is at 4 to 8 weeks post-fertilization, so still in the embryonic stage. But these are pre-placental tissue. Okay? So on the embryo on the left, you see that's an embryo blastocyst at 5 days. And a lump on the inside is called the inner cell mass. That's where human embryonic stem cells come from, and that's later becomes a baby, or that's where we all came from. The outer ring is trophectoderm, and that's later becomes the placenta. Okay? And in an ectopic pregnancy, what happens is the embryo is traveling down the fallopian tube, and it gets stuck, and it continues to grow until it bursts the tube. And if it bursts the tube, the mother may die of internal bleeding. And so when a mother experiences a sharp pain, she'll go to the ER, get scanned, and the doctor will go, whoa, this is bad. We have to take it out right away, otherwise you might die. And they perform an emergency salpingectomy where they go in and they ablate both sides of the tube and they remove the mass. And when they remove the mass, they throw it away. Right? So my father, who's a MD PhD, and he's a key opinion leader for OBGYN in Taiwan, he got his MD in Japan and his PhD at St. Bart's, the University of London. But he came back to Taiwan practice. And he was performing the salpingectomy in 2003, and he was thinking to himself, why are we throwing this stuff away? There's got to be amazing things in here, right? And so we sourced the cells from the preplacental tissue, and we hand the mass back to the pathologist who throws it away. And the reason why they throw it away is because that blastocyst on the left side typically needs to implant into the mother's endometrium or uterus by day 7 or day 8. But you don't discover an ectopic until 4 to 8 weeks after. So there are many weeks in between where that embryo has zero connection to the mother, no nutrients coming to it, so everybody understands that that fetus is already non-viable. So when they take it out, all they have to do is check with a pathologist, and they discard it. So what we have done is now source the earliest cells that you could possibly get on Earth without any ethical issues. Some characteristics, I'll go through these pretty fast. We have markers of all 3 germ layers. They're ethically sourced, they're scalable, they're immune privileged naturally, they are pathogen-free and chromosomally very, very stable, so it's a very stable DNA, and we have a large IP estate to protect it. We're able to push these cells out to 85 population doublings, right? So that's n to the 85. And if you use 100 million cells per dose, that's 8 times 10 to the 17 doses. That's more cells and more doses you could ever want to use, okay? Our cells naturally express HLA-G because think about it, the embryo is a recombination of the father's gene and mother's gene. So how come this thing's able to implant itself in the mother's endometrium without her immune system going completely crazy? Hey, this is foreign, right? Well, it turns out that HLA-G dampens down all of the mother's immune system so that the embryo can survive, attached to the mother. And it's the same function now that you can now take the embryo and give it to a surrogate mother to take it to term for you. Pretty amazing, right? Well, because of that, our cells have this characteristic. We already have 97 patents, 52 granted, and we have research use only cells as well as GMP cells, which we just completed last month, okay? And this is where I think I will probably contribute the most to our industry, okay? Because if you have RUO cells from the same donor as the GMP cells, I can send you RUO cells or research cells all day long, and as soon as you succeed in your research, you just have to come back and get the GMP line from me and go right into IND-enabling studies and into clinical trials, saving yourself a heck of a lot of time and money and documentation, okay? So we have perfect product market fit. You have the earliest embryonic stage stem cells without any ethical issues. We have a platform that provides scale and immune privilege. We've de-risked the FTO, or the IP issue, with our IP portfolio, and we have put together the CMC documentation so you can literally just go to the FDA with it. And we want to accelerate everybody's product time to market and really just make cell and gene therapies affordable to everybody, okay? We're working on Parkinson's disease and type 1 diabetes and cell therapy. Parkinson's disease with National Taiwan University, and Parkinson's disease, or type 1 diabetes with A-STAR in Singapore. And we are working with the NIA, a National Institute on Aging, on cell senescence, and hopefully driving back biological age with our exosomes, or extracellular vesicles. So that's so 2 in working on the back end, and 1 working on the front end. And my partner and colleague, Kotbi, will be presenting on Sunday at 12 o'clock. And he will be presenting some of this data on the cell senescence. So very, very exciting stuff. But more importantly, I want to make these cells and extracellular vesicles, or exosomes, available to all of you here, all of you. So if you're working in this industry and you want something to try that's new and the earliest, call me. I will get it to you, okay? This is how we're going to make my 5 years. So here's the QR code. Take a snap, email me, call me, message me, and I will send you the deck so you can have all the information to use at will. Thank you very much.